Hi, my name is Paul Valdmanis. I'm an assistant professor in the Division of Medical Genetics at the University of Washington in Seattle. And today I'm going to talk about mechanistic insight into tandem repeat expansions in amyotrophic lateral sclerosis. And I'm really excited to talk today about some of the ongoing projects in the lab identifying tandem repeat expansions and their role in evolutionary biology and neurodegenerative diseases, including ALS. Tandem repeat expansions are known to cause over 40 neurological conditions. They've primarily been identified using large multi-generational families, which helps identify a repeat segregated from one generation to the next. And as you can see here, repeats can be present in all parts of the gene, including 5' UTR, introns, coding regions, and so on. And these all can lead to various neurological conditions. However, the 40 repeats that are described here are just a subset of the hundreds of thousands of tandem repeats throughout the genome, many of which may cause or modify disease in the context of neurodegenerative disease. And one of the open questions about these repeats is, how do they expand? How do you go from a short repeat copy with 4 or 5 CAGs, as in this example here, to an expanded allele? Do the repeats expand at the beginning, the middle, or the end? And how many repeats are added at any one particular time point? And in some cases, it's not even the repeat itself, but it's actually the internal sequence that's much more critical in terms of disease pathogenesis. And so in this example, with this pedigree here, you have these individuals with over 1,000 copies of the pentanucleotide repeat. And individuals unaffected with spinocerebellar ataxia in this family have this pattern of a pentanucleotide repeat that is a pure track that's only in gray, and this is this ATTCT sequence. However, the 4 individuals with SCA in this family have this slippage from an ATTCT to an ATTCC, and this leads to the phenotype in this family. And the amount captured by whole genome sequencing is just a subset of the whole read. And I should point out that these are identified through long read sequencing approaches. So it emphasizes the point that as we're expanding the ability to sequence the genome using long read sequencing, we're identifying some of these repeat expansions that were previously hidden or intractable to short read whole genome sequencing. So with that in mind, we set out to identify potential tandem repeat expansions in amyotrophic lateral sclerosis, or ALS. And ALS is also known as Lou Gehrig's disease and is the result of the degeneration of upper and lower motor neurons in the brainstem, spinal cord, and motor cortex, leading to loss of muscle innervation, paralysis, and ultimately respiratory failure right at the heart of adult life. And no drugs are able to prevent ALS onset. A couple can delay progression by about a month or so, underscoring the need for better therapeutics for this devastating disorder. The genetic basis of ALS is starting to become clearer. There's about 5 to 10% of individuals with ALS that have a family history of ALS. So that is one or more relatives with the disorder as well. But as you can see here, some of the larger pedigrees with 10 or more individuals affected with ALS are the ones that are used to identify candidate genes and mutations or pathogenic variants in some of the diseases in ALS. And 4 genes have been identified for that account for the majority of familial ALS cases. And really the past 2 decades has seen tremendous progress in identifying these genes and other genetic risk factors. So the 4 genes are superoxide dismutase, or SOD1, TARDBP, which encodes the TDP43 protein, a gene called fused in sarcoma. And interestingly, the most common genetic culprit is a G4C2 hexanucleotide repeat expansion in the intron of a gene called C9orf72. And this accounts for about 40% of familial and 10% of sporadic ALS cases. And additional genetic contributions have been identified through whole genome sequencing studies and whole genome association studies and exome sequencing studies, revealing additional genetic culprits in disease. Often these are rare variants that are identified in a handful of individuals or families, or in the case of GWAS studies, point to loci that are now being examined in more detail to identify the causative variant at these regions. But as we're approaching 2020 and beyond, we're at this exciting time where we have these new technologies available to us, where we can identify novel risk factors, and in particular through single cell sequencing and long read sequencing, we can identify some of these regions that really could help explain some of the missing credibility in ALS. And so to kind of speak about our work, we started out with one of the families that initially had a FUS variant, and we'd taken a look at this family as well and performed some genome-wide screens to try to find if there was an additional genetic modifier for this region. And as you can see, some of the individuals that are white, indicating they haven't developed disease yet, still have the variant, indicating that perhaps there's another second hit or another genetic modifier in this region. So from kind of old school linkage studies, we'd actually mapped this second region in this family to a region on chromosome 18, fusion sarcomas on chromosome 16. And in this chromosome 18 region, all affected family members share a 4 megabase segment of DNA. And the genome-wide significance of this linkage was quite significant. So we took a look within this region to see whether there were other risk factors that could help explain some of the missing additional genetic risk factors in this family. And this is just to kind of point out, by using both this chromosome 18 locus and the FUS variant, we could start to explain some of the individuals that had developed ALS but hadn't, that are shaded in black, but did not perhaps have a FUS variant. And this kind of like combination is best exemplified by these 4 siblings here, where you can see they have all 4 possible genotypes, either the FUS variant, neither variant, the chromosome 18 locus, or both, and it was only the ones with both that had that developed disease. And so in our search, we came across this human-specific tandem repeat expansion that each copy of the repeat is 69 base pairs. And so this is in the last intron of a gene called WDR7, and each copy of the repeat actually folds into this really interesting hairpin-like structure where there's regions of self-complementarity and then a loop region and then a gap and then another hairpin. The reference genome has about 6 copies of this repeat, but actually individuals in that family had over 30 copies of this repeat. Again, when this repeat is inserted into RNA folding algorithms, it reveals this really unique hairpin structure of this intronic sequence. So the gene WDR7 itself encodes a protein that localizes to synaptic vesicles involved in the control of calcium-dependent exocytosis of neurotransmitters, as well as acidification of endosomal vesicles. So all of these are relevant to ALS etiology. So we wanted to take a look further at WDR7 function and kind of the evolution of this repeat expansion. What really surprised us was that this is almost exclusively a human-specific repeat. It's not present in any lower mammals, including lemurs and other non-primates. Old world monkeys, starting with the squirrel monkey, start to have about a 47 base pair region of the repeat. So it's about 2/3 of the repeat start to appear in these genomes. By the time you get to old world monkeys here, starting with the orangutan, you actually jump to 1.5 copies of the repeat. And by the time you get to humans, you have 6.5 copies up to 33 copies in this ALS family. So we could track the progression and the expansion of this repeat throughout primate evolution, But we were also interested kind of beyond this role, you know, what is this a risk factor in ALS beyond just the family, essentially? And so to do this, we were curious to take a look at individuals with sporadic ALS, the large cohorts, and try to identify the repeat size to see if we could find additional individuals with high repeat copy number. And we were really fortunate in that we were able to actually PCR amplify this repeat. We were able to design on the left here in orange primers either at the beginning or out at the end of the repeat. And we could actually then sequence through various copies of the repeat. And shown on the left is 4 copies of the repeat, but on the right is a gel of a PCR, and you can see each of the rows is a different individual with often 2 alleles of different lengths. So we were able to actually use this information to PCR amplify and run on a gel hundreds of ALS cases and controls from the Coriell cell repository of bank DNA samples. And when we did this, we actually found an enrichment of repeats that were 30 or more in length in individuals with ALS relative to controls. We found cases where there perhaps was like a premutation just below that threshold. And we also found one individual at the far right in orange with 86 copies of the WDR7 repeat. This is a female without a family history of ALS who died at the age of 72. So combining all of these samples and just measuring the largest or the longest repeat in each, we actually could see that there was a shift to longer average repeat in ALS samples to 17.7 copies versus 15.2 copies in controls. And what we were able to do was then use this information to, and confirm this information using 2 additional populations. And we used whole genome sequencing and read depth to estimate copy number in 2 cohorts, one from Answer ALS and another in samples from Quebec, Canada. So in our mind, even though there's control individuals have high numbers of, or high repeat copies as well, there's this shift, there's this kind of modifying effect in the context of ALS. The second thing we wanted to do, and this relates to the spinal SCA pentanucleotide repeat that I was showing earlier, was to determine whether there was any internal repeat sequence that was different in ALS. So to do this, we took those same primers that we used to amplify WDR7, but we added barcodes to the 5' and 3' end. And combinatorial use of those barcodes allowed us to actually PCR amplify 144 individuals or 288 alleles in one lane of tactile long-read sequencing. And we had a problem, though. And the problem was that how do we then take all that information and encapsulate it and be able to align it to identify if there was enrichment? And one of the examples here is that 86 copies of the repeat where you see have over and over again that repeat expansion through the genetic sequence. And we needed to find a way to kind of align that along with some of the other ALS individuals and controls that we had that underwent long-read sequencing. So we used a couple of tricks. And the first thing that we wanted to do was to look at the potential internal sequence at each repeat unit. So again, the WDR7 repeat is 69 nucleotides in length. And of those 69 nucleotides in color, you can see if there's a dot, it's the same as the consensus sequence. If there's a color, it means there's a base pair change. And there's 6 positions where there are base pair changes throughout the repeat. And so what we did was we converted this to a single color for each of the 16 most common repeat sequences that we observed. And you can see each one has a different combination of these internal 6 variants that are present. When we did this, we found that there wasn't actually a difference between each of the internal sequences between ALS cases and controls, but we could still use this information to try to design a pattern and convert some of this information to a kind of a color-coded map. So again, from each individual, we identified the start of the repeat, we aligned this, placed each repeat into a separate row, converted this actually to a single letter code to avoid some of the complexities of this information, and converted that code to a color. And the reason why we chose these single letter codes is that we could actually use amino acid clustering alignment programs to align some of the repeats to identify patterns in their structure. And then the colors helped us visualize things rather than a series of letters together on the slide. We can kind of like immediately kind of your eye is drawn to some of the patterns in this sequence. And so this is a complex plot that we put together, but what you can see here is that the repeat length is here on the y-axis and individual alleles are on the x-axis. And starting on the left, you have about 8 or so alleles, individuals with, 5, 2, 3, 4, and 4 copies of the WDR7 repeats for each of them. We continued to do this iteratively, and when we were able to put it together, we saw this complex pattern of the repeats. But we noticed something, and we were always kind of sequencing this from the 5' to 3' direction. But in fact, the 3' region of the repeat had a much more similarity in terms of the colors that were present. And so when we realigned these on the 3' end, we started seeing these amazing patterns and clusters of groups of individuals that each had the same, a similar combination of the WDR7 repeat. And these were often very much fixed at the 3' end, but then started to diverge at the 5' end. And there were several kind of different original clusters. You can see ones with a number of dark blue reads, numbers with yellows and purples and dark red. And as every time we kind of looked at this repeat, we learned more information. We started noticing patterns that were emerging from this data. One of the first things we noticed was that there seemed to be this kind of biphasic kind of introduction to the repeats. So the repeats weren't showing up one at a time, but often in groups of 2. And you can see that, especially in the middle of this repeat, where there is this dark red and light purple clusters of repeats. And so when we analyzed these iteratively across all of the 288 alleles from 144 individuals, we saw this kind of periodicity of the repeats, where you were more likely to see repeats, the same repeat, 2 repeat units from each other, 4 repeat units, 6, 8, and so on, than you were to see the repeat unit immediately adjacent to itself. So there's something about the 2 repeat units together that precipitates some of this expansion. We could see large duplication events in some of the cases. You can see this group of about 6 repeat units that were copied several times in the same individual. And we can also see that, again, speaking to the 3' to 5' directionality of the repeat, whenever we saw a new repeat unit and one that wasn't part of that original 16, these ones we would color in black, and these ones would appear right at the 5' end of the repeat. And not only would they appear once, they would actually, if they appeared a second time, like this yellow-black segment, they actually expanded in combination with that previous repeat unit. Again, suggesting there's something unique about this 138 or so nucleotides that was important for this expansion event. The initial sequencing run that we had performed was actually selected for individuals with 20 repeats or more, because we were really concerned about being able to amplify some of that internal sequence with the really large repeats. Once we started seeing those patterns, we then went and repeated the PacBio long read sequencing run with a second cohort of individuals. And the second cohort had additional populations, both with AD and control, but also individuals from the 1,000 genomes population, allowing for kind of a more diverse cohort of individuals looking at repeat expansion. What we noticed, because we didn't, and in this case we didn't size select, so we were noticing several alleles on the left that were of the shorter nature, but we also saw that exact same pattern that we saw before, where you had these clusters of the same kind of segments of repeats, indicating that the alignment that we put together was reproducible. And in using some of these 1,000 genomes populations samples from worldwide populations, we could start to see patterns emerging from some of the groups. So for instance, I've boxed here samples from Africa on the left and samples that were outside of Africa that had the kind of the same original base structure, and then halfway through there's this divergence point between pink and purple alleles, and African samples maintain their trajectory from there on in out, and non-Africans have different alleles that are added, suggesting that perhaps there was an evolutionary time point when this breakpoint occurred. And we saw the same phenomenon in repeat expansions that were specific to Han Chinese individuals, where they had expanded using their own combination of these internal repeat sequences. So we think this is a fascinating kind of tool that can be used to kind of track evolutionary expansion of some of these tandem repeats across different populations. From a multi-generational standpoint, and within one pedigree, we can use the information to, again, track segregation from parents to children. And because we can combine this with the internal sequence information, we can actually find individuals with similar length alleles, but actually different internal sequence within them. And then another cool thing that we could do was then, because we had all this information that we gathered from long-read sequencing, we could go back to short-read sequencing and whole genome sequencing to try to determine and predict what alleles were present in certain populations. And in particular, we were interested in ancient populations like Denisovans and Neanderthals. And so as you can see here, this is an IGV plot where the top panel is the alignment of short reads to the WDR7 intron, and in the middle is the region with the VNTR. And you can see there's a very strong pileup of short reads at that VNTR itself, suggesting that it's much larger than the reference human genome, which has 6 copies. And conversely, Neanderthals have more reads at the surrounding genomic region, but a paucity of reads at the VNTR itself, suggesting that perhaps it is a shorter repeat than in the human reference genome. And in fact, we can actually go and identify what, because of our, with that internal sequence, what the actual repeat units are. So we can see that there are 4 different repeat units present in the Neanderthals. And so they either have a combination of 1 and 3 copies or 2 and 2 copies of the repeat. And then Denisovans, we can map the number of repeats and the number of counts of repeat copy numbers, and actually use this information to try to reconstruct potentially their ancestral allele at the WDR7 locus. And beyond this one repeat, we were really interested, can we generalize this information across the 55,000 variable number tandem repeats in the human genome, and a variable number meaning repeats with internal sequence composition 7 nucleotides or greater. And so for this, we took advantage of a couple of publications from the group of Evan Eichler, including this one here that maps variable number tandem repeats across the genome. And we can see there's a lot of them in green that are that cluster around telomeres or subtelomeric regions, but we also had enough within genes and around genes that we could extract the information and take a look at whether they expanded in a manner similar to the WDR7 repeat. And our selection criteria were ultimately around 400 repeats that are human-specific with a single site in the genome and a repeat unit 25 nucleotides or greater. And what we were able to do was take advantage of 15 individuals, again from the 1,000 genomes population samples, that had undergone long-read phase sequencing. And these individuals had, aligned here on the y-axis, had various copies of some of the repeats that we were taking a look at. And so each kind of, each row here represents a different individual and their combinations of repeat length. In this case, for a 41 nucleotide repeat unit in the intron of a gene called NWD2. And as you can see, when we kind of took a look at some of the repeat structure in the organization, this is kind of what we were expecting to see, is that, again, similar to WDR7, we color-coded each of the different repeat units. So in light blue, you have one sequence that is repeated several times, And in purple, you'll have a repeat unit, again, of that 41 nucleotides with maybe a base pair change somewhere throughout it. And we could see both combinations of the light blue and purple throughout the repeat, and lengths that span between less than 10 to over 40 copies of that repeat. So again, it suggests to us that WDR7 is not alone in terms of its variability, extreme variability in terms of length. However, this repeat itself was much more pure in terms of the internal sequence. There were fewer nucleotide variants than the 16 or so we observed in WDR7. When we examined additional repeats, VNTR repeats, we found other categories of kind of dormant repeat units. In this case, again, this is a 41 base pair repeat in a gene called LOC 102.725.191 that primarily had about 30 copies of this repeat. You can see there's a lot more colors that are present. That means that there are a lot more internal nucleotide variants at each copy of that 41 base pair repeat unit. However, whatever expansion was taking place and the nucleotide changes that were taking place throughout that expansion were occurring much earlier on. So the repeat had seemed to have become fixed in modern humans. And a third category of repeats that we observed, which was probably less than 10% of the time, but we saw other repeats where there were different alleles that were present and major alleles that were present that had different combinations of length and internal sequence. So the first three individuals here had one set of colors indicating kind of what a fixed kind of group of alleles, and then the bottom set had another cluster, and in the middle you actually had this individual that had a kind of a switch from one allele to the other, so some kind of recombination event. So these were the three kind of main categories of tandem repeats we observed when looking at long-read sequencing. But we also found some really interesting outliers and unique kind of nuggets in terms of their properties. And in this case, this is a 33 base pair repeat unit in the 3' UTR of a gene called VPS53, and it has three separate segments within its internal tandem repeat. So the first segment is fixed in length, but variable at each copy of the repeat. So each copy of the repeat had a different internal sequence. And then you had a second segment where there was mostly a pure sequence, but some variability in length, apart from these few individuals with a kind of a darker blue color. And then you had this really interesting third segment where there were combinations of three repeat units together that were expanding, and quite a bit of diversity in terms of the repeat length. So that segment 3 is the one that seems to be actively expanding, and one that we're really interested in determining, as we identify more individuals, what accounts for some of this variability in length, and why is it expanding, again, in combinations of 3 repeat units, or about 100 base pairs, versus the WDR7 repeat that seems to be expanding in 2 repeat copies. And we could find, again, and we could really kind of use this information to then narrow down exactly the components involved with repeat expansions as we kind of analyze more and more individuals using long-read sequencing. So again, this is a repeat in a gene called SLC22A1, where you have a bit more than 40 copies of that repeat in some individuals. And you can see here there are gaps. So these gaps in white represent kind of deletion events that are present, that are the deletions that are present in certain individuals. We also have duplication events. So you can see starting with orange up until purple and light purple colors. This whole block gets duplicated in a series of individuals. And we can see once that first duplication event takes place, we actually see a second duplication event in this individual up here, because now the orange allele has been placed next to the light purple one, and you can see a second event where they've added another segment of about 6 or 7 repeat unit numbers at the top there. So you can kind of use this to progressively track how a repeat is expanding amongst these individuals. And then finally, using whole genome sequencing, we could actually identify regions much larger and much more complex repeats that differed in length in hundreds of repeat copies. And so to do this, we took a look at short read sequence data, identified repeats that were much longer in length in, for instance, African samples versus admixed American samples. And Prop1 is an example of a repeat. In this case, actually, that's longer in African samples. Art1 is a repeat that, on average, has a much longer length in admixed Americans. And then we could, using long-read sequencing, identify, again, that internal sequence and of each repeat unit and the colors that represented some of the internal variability in these alleles. And so a number of the individuals from the African samples were appearing at the top of this chart where they were only blue, and at the bottom were samples from European and admixed Americans where you had these expansions in combinations of 2 kb blocks. So as we're getting more and more long-read sequencing information, we can get a good idea of how these repeats are expanding, the internal sequence that is combined along with this, and understand kind of some of the differences amongst humans. So in summary, I want to say that we're really interested, and we truly believe that long-read sequencing of tandem repeats can provide a lot of information about both human evolutionary events, but also risk factors for neurodegenerative disease. And we believe that VNTR expansions, tandem repeat expansions in ALS can represent novel disease risk factors, not only in ALS, but also in other neurodegenerative diseases as well. And further analysis of the internal sequence of these repeats can help reveal mechanisms of tandem repeat expansion. So with that, I'd like to thank members of the lab, past and present, as well as funding sources and our collaborators at the University of Washington and McGill University. Thank you very much.